It's pretty wild when you think about it. Insulin, we hear the word all the time, but its story. Even finding versions in things like cone snails, using it as a weapon.
Yeah, it's amazing, isn't it? Like chemical warfare for snails.
Hmm.
And it really challenged those early assumptions that hormones would all be these tiny molecules. Insulin's quite large, relatively speaking.
A big protein doing this fundamental job. It highlights just how deep its biological roots go.
Yeah, exactly. That cone snail example. It
Mm.
just drives home how essential this molecule is, way beyond just humans. It's ancient.
Absolutely. And that's actually a great place for us to start today. Welcome back to the deep dive. We are really going deep on insulin this time.
We are. And for this, we've pulled together quite a range of sources for you. We're looking at, the core science from research papers, medical guidelines, but also regulatory info, historical accounts, trying to get that full picture.
Right. So for you, the learner, our mission here is to unpack this really critical hormone. We want you to walk away with a solid understanding of where it came from, how it works, how it's made, which is fascinating in itself, the rules around it, the controversies even, and just its massive impact.
Okay, so let's jump right in. What is insulin,
How does that
fundamentally?
work?
Well, at its core, it's what we call a peptide hormone. So basically a protein built from amino acids acting as a messenger. And it's produced by very specific cells, the beta cells, in the pancreas.
In those islets of Langerhans.
That's them, little clusters within the pancreas.
Oh, yeah.
And their main job, the big one, is managing your blood glucose, your blood sugar.
Okay, so how does it do that? What's the mechanism?
It works in primarily two key ways. First, it basically tells cells in your body, thick muscles, fat tissue, hey, soak up glucose from the blood.
Opens the door, sorta.
Exactly. And second, it signals to your liver to put the brakes on making more glucose. So it reduces glucose coming into the blood and increases glucose leaving it. It's a balancing act.
Sounds pretty sophisticated. More than just flipping a switch.
Oh, definitely. And the molecule itself, it goes through a whole production line before it's ready.
Yeah.
It starts as this inactive precursor, preproinsulin.
Okay.
Then inside the cell, in an endoplasmic reticulum and Golgi apparatus, think of them as the cell's factory and processing plant. It's modified, bits are snipped off, it folds correctly.
And you end up with the final active insulin.
Right. The active form has two chains, A and B, linked together by disulfide bonds. And interestingly, when a piece called the C-peptide is cut off to make active insulin, it gets released too. We can actually measure C-peptide in the blood to see how much insulin the body is making itself.
Ah, that's clever. A useful byproduct. And you mentioned earlier this basic structure. It's found across lots of different animals.
Yes, the sequence of amino acids, the blueprint, is remarkably similar across species. It's highly conserved, evolutionarily speaking, which just, hammers home how vital it is.
So even though, say, cow or pig insulin isn't identical to human insulin,
Right, there are small differences, 3 amino acids different in cows, 1 in pigs, but they still work well enough in humans. It shows the core function is preserved even with minor tweaks.
okay, so this hormone is crucial for keeping blood sugar stable. What happens when that system goes wrong? When you don't have enough or it's not working properly?
Yeah, that's when we run into problems. Primarily, high blood sugar hyperglycemia, which is the hallmark of diabetes.
And there are the two main types we hear about, type 1 and type 2. How does insulin play out differently there?
They are quite distinct. In type 1 diabetes, it's an autoimmune issue. The body's own immune system mistakenly attacks and wipes out those beta cells in the pancreas.
So the factory gets destroyed, basically.
Exactly. The result is an almost complete inability to produce insulin. It's an absolute deficiency.
Okay. And type 2 is different. Less about destruction, more about malfunction.
It's generally more complex, yes. There can be some beta cell loss in type 2, maybe linked to things like amyloid protein buildup in the islets, but it's usually not the main driver, and it's not typically autoimmune.
So what is the main issue then?
Often it's a combination. The remaining beta cells might not secrete enough insulin when needed, and crucially, the body's tissues, muscles, fat, liver become resistant to the insulin that is there. It's like the locks are getting rusty, the key doesn't work as well.
So the signal isn't getting through properly.
Precisely. Plus, in type 2, there's often another issue. The hormone glucagon, which raises blood sugar, isn't properly regulated. It keeps getting released even when blood sugar is high. So you've got multiple things pushing blood sugar up.
Wow. Okay. So it's not just about less insulin. It's also about sensitivity and other hormones getting involved.
That's a good way to put it. And beyond type 1 and 2, you have other conditions too, like insulinomas, rare tumors that make too much insulin, causing low blood sugar. And things like metabolic syndrome or PCOS are often linked with insulin resistance.
It really is central to so much of our metabolism. Now, obviously, we haven't always had insulin available as a medicine. It has this incredible history of discovery, right?
Oh, absolutely. A long and fascinating journey. People knew something was wrong with the pancreas and diabetes way back in the 19th century. But isolating the active substance? That took time.
The early 20th century was key, wasn't it? Banting and Best and McLeod.
Yes, their work in the early 1920s, leading to the first successful treatments, was monumental. But the scientific understanding kept evolving rapidly after that. John Jacob Abel crystallized insulin in 1926, a huge step for studying its chemistry.
Getting a pure form.
Exactly. And researchers like Somogyi, Doisy, and Schaffer proved it was definitely a protein around 1924. Then Jensen and Evans Jr. identified key amino acid components like phenylalanine and proline in 1935. Little by little, the picture got clearer.
But figuring out the entire sequence, every single amino acid in order, that sounds incredibly difficult for the time.
It was a Herculean task.
Oh.
Frederick Sanger finally cracked it in 1951, determined the full amino acid sequence. Groundbreaking work earned him a Nobel Prize.
Oh, once they knew the sequence, could they make it?
Well, that was the next big challenge. Synthesis, making it from scratch in the lab. Two groups, Ketsianises and Zons, managed it pretty much simultaneously in the mid-1960s, and Chinese scientists also synthesized bovine insulin around then.
And then seeing its actual 3D shape.
That came in 1969. Dorothy Hodgkin used X-ray crystallography to map out its complex three-dimensional structure. Another Nobel Prize-winning effort. It took decades of brilliant work from many people.
An amazing scientific story. And for a long time, the insulin people used actually came from animals.
That's right. For many, many years, the source was purified insulin from the pancreases of cows and pigs. It saved countless lives, absolutely, but it wasn't identical to human insulin, and sometimes it could cause immune reactions or allergies.
Which leads us nicely into how insulin is used clinically today. What are the main reasons someone would need insulin therapy?
Well, the most obvious is type 1 diabetes, where the body makes virtually none. Insulin replacement therapy is simply essential for survival there.
No question. But it's also used quite a lot in type 2 diabetes, isn't it? Even though the body might still be making some.
Yes, absolutely. In type 2, treatment usually starts with lifestyle changes, maybe metformin or other oral drugs. But if blood sugar targets aren't met, or if someone presents with really high symptomatic blood sugar, then insulin is often the next step.
How is it typically started in type 2?
Often, the first step is adding a basal insulin. That's a longer-acting type designed to cover the body's background insulin needs overnight and between meals. People usually continue their other medications alongside it.
And then sometimes more insulin is needed.
Yes. If blood sugar still spikes after meals, a bolus insulin might be added. This is a rapid-acting type taken just before eating to cover the glucose coming in from food.
We hear about different kinds, like analogs. What are those?
Insulin analogs are basically versions of human insulin that have been tweaked, genetically engineered, to change how quickly they work or how long they last.
To make them work better for certain situations.
Exactly. Rapid-acting analogs, for instance, kick in faster and wear off sooner than regular human insulin. That can be really helpful for matching insulin delivery to meals, potentially reducing lows later on.
And the long-acting ones?
Long-acting analogs are designed to provide a steadier, peakless background insulin level over, ideally, 24 hours. This predictability is a big reason why analogs, both rapid and long-acting, have become so common, especially in type 1 management.
Makes sense. And how people take insulin has evolved too, right? It's not just vials and syringes anymore.
Definitely not. Insulin pens, pre-filled devices that make dosing easier and more discreet, are hugely popular.
Yeah.
And then you have insulin pumps.
Right, the ones that deliver it continuously.
Yes, small devices worn on the body that deliver insulin through a tiny tube under the skin. They offer a lot more flexibility and fine-tuning. And now we're seeing really exciting advances in automated systems.
The artificial pancreas type systems.
Exactly. Hybrid closed-loop systems, sometimes called the bionic pancreas. They link a continuous glucose monitor with an insulin pump via a smart algorithm. The system automatically adjusts insulin delivery based on real-time glucose readings.
That sounds like a game changer, especially for type 1.
It really is proving to be. The real-world data and patient experiences are incredibly positive. It takes away a lot of the constant calculation and burden.
Amazing progress. Okay, let's shift gears a bit. How is this stuff actually made today? I assume it's a far cry from grinding up animal pancreases now.
Oh, completely. The vast majority of insulin produced today uses recombinant DNA technology. It's a biological manufacturing process.
Which means it's different from making a typical chemical drug.
Yes, very different. Because you're working with living cells and complex biological processes, there's inherent variability. So quality control, risk management, they have to be incredibly stringent every step of the way. It's not like just mixing chemicals A and B.
So what are the general steps if you're making a biologic like insulin?
Well, the WHO lays out general guidelines. You start with your source material, the engineered cells. You process them, create carefully controlled cell banks, master banks, working banks to ensure consistency.
Like a library of the producer cells.
Sort of, yeah. Then you grow these cells in huge batches, in bioreactors, under very specific conditions that's cell culture or fermentation. They produce the protein you want, like proinsulin.
And then you have to get the insulin out.
Right. That's the purification stage. Very complex, multiple steps to isolate the target protein and get rid of everything else, cell debris, other proteins, etc. Then it might be formulated, maybe combined with other things to stabilize it or adjust its action profile, and finally filled into vials or pens.
You mentioned recombinant DNA. We often hear about E. coli bacteria being involved. How do they fit in?
E. coli is a real workhorse for this. Scientists engineer these bacteria by inserting a piece of circular DNA, a plasmid, into them.
Okay.
And this plasmid contains the human gene, the instructions for making either the A and B chains of insulin separately or the single-chain proinsulin precursor.
So the bacteria become tiny insulin factories.
Essentially, yes. They're grown in large tanks with nutrient broth. Often, an antibiotic like kanamycin is added. The plasmid also contains a gene for resistance to that antibiotic.
Ah, so only the bacteria that successfully took up the insulin-making plasmid survive.
Exactly. It's a selection mechanism. Then later, techniques using special enzymes, restriction enzymes like BAMA-high, are used. They cut the DNA at specific points.
Like molecular scissors.
Right. And by looking at the pattern of DNA fragments produced, scientists can confirm that the insulin gene is present and correct in the E. coli colonies they've selected. It's like checking the genetic blueprint is right before mass production.
Fascinating. A real blend of biology and engineering.
Yeah.
Now, what about the rules, the regulations around insulin? Has that been straightforward?
Not always, especially in the U.S. There's some unique history there. A very early step was the 1941 Insulin Amendment. It actually required the FDA to batch test insulin for purity and potency. That was quite forward-thinking for the time.
But it wasn't treated like other biologics for a long time.
That's the key point. Historically, insulin in the U.S. was regulated under the Food, Drug, and Cosmetic Act, not the Public Health Service Act, like most protein therapies, most biologics.
And that difference mattered.
It did, particularly for the approval pathway for biosimilars, those highly similar, potentially more affordable versions. They had to use a different, arguably more complex pathway, the 505(b)(2) pathway, instead of the dedicated biosimilar pathway, 351(k).
But that's changed recently, or is changing.
Yes, the transition happened around 2020. The idea was to bring all biologics, including insulin, under that same Section 351 umbrella in the PHS Act. The hope was to streamline biosimilar approvals.
Okay, so what are biosimilar insulins and what are the hurdles? You mentioned affordability.
Right. A biosimilar is a biologic drug shown to be highly similar to an existing approved biologic, the reference product. No clinically meaningful differences in safety, purity, and potency. Because they come from living systems, they aren't exact identical copies like small molecule generics.
So getting them approved requires showing they're really, really close.
Exactly. And a key concept, particularly in the U.S., is interchangeability. This is an extra designation. It means a pharmacist can substitute the interchangeable biosimilar for the original brand name biologic without needing to check with the prescriber first.
Like how generics are substituted now.
Pretty much.
Oh.
But achieving that interchangeability designation requires additional studies and data, and it's a high bar set by the FDA. It's seen as crucial for driving real market competition and price reductions. The approach differs elsewhere, though the EMA in Europe, for example, doesn't make that specific interchangeability call at the EU level.
Which brings us to the elephant in the room. The cost. Insulin prices, especially in the U.S., are notoriously high.
They are. It's a huge issue. Despite insulin being a century-old discovery, the U.S. market is dominated by just 3 main companies. There hasn't been the kind of robust biosimilar competition that many hope for, or that exists for other drugs.
And that lack of competition is seen as a major driver of the high prices.
It's certainly a huge factor. Prices in the U.S. are dramatically higher than in Canada, Europe, other developed nations. It's led to significant access and affordability crises for many people.
Have there been efforts to figure out why this is happening? beyond just lack of competition?
Oh, yes. There's been a lot of scrutiny on the whole supply chain, particularly the role of pharmacy benefit managers, PBMs. These are the intermediaries between manufacturers and health plans. Investigations by Congress, the FTC, they're looking into the rebate systems and business practices to see if they contribute to inflating list prices while maybe benefiting the middlemen.
Complex stuff. Are there solutions being talked about?
Well, several things are on the table or have been tried. Allowing importation from countries like Canada is one idea, though it has its own complexities. There have been state and federal legislative pushes to cap copays or somehow control prices. And of course, encouraging more biosimilar entry and uptake is still seen as key.
How does this compare globally? Are other countries handling biosimilar insulin regulation differently?
It really varies. The EU, Japan, Australia, Canada, they all have established pathways, maybe with slightly different data requirements or approaches to interchangeability. Some places, like Brazil, even have multiple tiers of biosimilar pathways. And you see a growing trend in countries like Brazil, Cuba, Iran, China, to develop local manufacturing capacity, sometimes through partnerships. It's often driven by a desire for better access and lower costs.
And patents must play a role in this competition landscape, too.
Absolutely. Like with many drugs, companies use various patent strategies, sometimes called patent thickets, on the drug itself, the delivery devices, manufacturing processes. This can significantly delay the entry of biosimilar competitors long after the main patent on the molecule has expired.
So this whole regulatory picture, the competition issues, it all has a massive economic impact, doesn't it?
Huge. Insulin represents a really significant chunk of healthcare spending, especially with the high U.S. prices. The potential savings from wider biosimilar use are substantial, theoretically.
And the price difference drives things like people traveling to buy insulin.
Yes, unfortunately. The stark price differences between the U.S. and, say, Canada or Mexico lead some individuals to travel across borders just to afford their medication. It highlights the desperation. There's also ongoing debate about the cost-effectiveness of the newer, more expensive analog insulins versus the older, cheaper human insulins, especially for some people with type 2 diabetes, where the clinical benefit difference might be less pronounced than in type 1.
Beyond the dollars and cents, though, insulin's impact on, well, on culture and just daily life for people with diabetes, it's profound.
Completely transformative. You have to remember, before insulin, type 1 diabetes was fatal, usually very quickly. Insulin turned it into a manageable, albeit challenging, chronic condition. It gave people their lives back.
And even today, the type of insulin regimen someone uses can really affect their day-to-day experience, can it?
Definitely. We look at patient-reported outcomes, or PROs. How satisfied are people with their treatment? How does it impact their quality of life? Studies comparing, say, basal bolus injections multiple times a day versus using simpler premixed insulin show varied results. Things like convenience, fear of needles, complexity, they all play a role in how people feel about their therapy. And sometimes responses can even differ across ethnic groups.
And just sticking with insulin therapy can be hard. Adherence is a big issue.
It really is. Particularly in type 2 diabetes, a surprising number of people don't take their insulin as prescribed consistently. Or they stop taking it altogether. That's called persistence.
Why is that? Is it just the injections?
That's part of it, for sure. The frequency, the timing, the planning involved, it can feel like a huge burden. But it's also about perceptions. Some people might see starting insulin as a personal failure or a sign their condition is getting much worse. Healthcare provider communication is critical here, understanding those patient perceptions, explaining the benefits clearly, addressing fears. Building that trusting relationship is key.
It sounds like managing insulin involves so much more than just the medicine itself. It's psychological, behavioral.
Absolutely. It requires significant patient education, ongoing support, and a collaborative approach between the patient and their healthcare team to overcome those barriers and achieve good long-term outcomes.
It's truly remarkable. We've gone from, a mysterious substance in the pancreas to these incredibly sophisticated delivery systems and manufacturing processes, yet we're still grappling with issues of access and cost a century later.
It's that contrast, isn't it? The incredible scientific progress alongside the persistent societal challenges. Insulin embodies that, perhaps more than any other medicine.
So, thinking about this whole journey, this deep dive, its history, the science, the economics, the human impact, what's final thought for our listeners to chew on?
Maybe this. Considering insulin's century-long history and how fundamental it is, what does the future hold? Not just for making better insulin or better delivery systems, but for ensuring everyone who needs this life-saving medicine can actually get it and afford it wherever they are in the world. That seems like the next big frontier.